to this point, because when it comes to nano formulations, the situation is strikingly different in Europe vis-à-vis the United States. Because if there's published data which shows that research in nano compound is very strong in the small and medium enterprises in the United States. I do not remember the name of the guy who published, but it's a published paper. If you want, I can dig out where I found it. Whereas the same amount, let's say 90–95% in Europe, is done by academic centers. So you see there is an asymmetry in terms of what is available in Europe in terms of openness to publish results and to make things known, vis-à-vis of the restraints in the United States which are linked to commercially confidential information. We are sort of blessed in Europe as we have an academic kind of backbone supporting development of such products. This is one of the advantages. Of course, we have a lot of disadvantages because these products never get to the fruition because there is no the industrial mentality to get into fruition. It's more on the learning, the discovering, the maybe IPs and patent, etc., rather than true progression into an actionable drug. But then I see, and we discussed with Frank yesterday, we maybe put together efforts, centers of excellence in the United States, prominent research groups in Europe, and we can create really a very strong synergy, also with colleagues from China. They've done fantastic work from India, from Switzerland. There's a lot of things we can put together.
Switzerland, I don't know, but...
Well, you're the big guy.
Oh, this is taped, isn't it? Can you take that out, please? But Gerrit, you can correct me, as usual. Thanks, Frank. Thanks for the presentation. Thanks, Marissa, for your comments. There will definitely be more time to discuss these issues later on. Yes, but the light is flashing already for a long time. You can't see that, but I think there's something going to hit me if I really stop here.
I will take it later.
Yeah, there is a panel discussion, so don't worry. There's plenty of time then. But I really want to move on, and it is a real pleasure to introduce or to ask Scott McNeil to come to the forefront, because he may be different than what Marissa was saying, that it's all company work that's being done in the States. There's also some other work going on there, and which is published normally. So, Scott, please take the floor and explain to you what the NCL is doing.
Okay, thanks, Don. So you've been hearing about characterization methods from us for the last 4 years and several talks here during this conference on how complicated characterization is. And you heard the definition of NBCDs. It's a drug complex or formulation that cannot be adequately described just by physical chemical traits. So for example, in a multifunctional nanoparticle, we can do exquisite characterization, and I can tell you averages of how big that is, first principles. I can tell you components of each one of those and their distribution. But take something like this, where there's a formulation that each and every time that formulation is made, each batch is differently, even under GMP conditions. So compound that and try to put yourself in a regulatory position as these two are required to do. And what do you have to be able to make comparisons between those two components or those two formulations? Yes, they may have the same API, but do they necessarily have to be exactly the same size or have exactly the same amount of polyethylene glycol, especially when the original product has quite a bit of variability as well. So I refer to this as a two-bucket approach. We have, in the US, 2 buckets to be able to evaluate nanomaterials, especially for the follow-ons. Well, for the follow-ons. One of those, as you're seeing here, is the generics pathway, where we can, in fact, define that product by PCC traits. We can say with good accuracy what the mass spec is, the purity, the excipients, and so forth. Under this within the US system, there's not a mandated in vivo study if they can show equivalent comparison. This is where we get things like monographs published by USP. We can have a recipe where it's step-by-step production of that material. But then it gets very complicated when you have something like a biomolecule, where yes, we might be able to have the same sequence, the primary sequence of amino acids for that product, but things like glycosylation on the surface are going to vary from cell line to cell line, even batch to batch. So with this, because of that variability, there's a requirement often to do an in vivo comparison study for a follow-on product. But what about nanomedicines and what about NBCDs? Well, we're stuck in the middle between those two. Some of the drugs may favor a small molecule such as Doxil. It's a reformulation of a small molecule. And then there's Abraxane, where there's pretty good evidence that the carrier, albumin, contributes to the efficacy and the mechanism of action. So then how would you do a comparison of Doxil or Abraxane versus a follow-on that may not use the same excipients or same components to get there? Let me share some of our research that we're doing at NCL to address some of these things, where just simply comparing PCC attributes is not going to be a true comparison of things like efficacy or drug release. What you see there in blue is an encapsulated drug in a nanoformulation. Keep in mind, and we have quite a bit of empirical data on this at NCL, that the drug, which you're seeing as Ds there, exists in several forms. It exists in a free form, and it exists in a protein-bound form, and it exists in an encapsulated nanomaterial form. Each of those have different traits associated with it, but it's actually the free drug that is the main toxicant or the main drug piece of action within the cell. And so being able to characterize those, we're finding that if we can truly characterize and quantitate the free drug, we're far easier, it's far easier to make comparisons between nanomaterials and follow-ons that may not have anything to do with the physical properties of the original drug. For example, I'll show it to you here. The graph that you're seeing there is a log graph where the free and unbound is orders of magnitude less than the total drug. We have very good methods for monitoring total drug, especially when we do comparisons, but very few to monitor the free drug. Protein bound we can do fairly straightforward. So we are now aggressively pursuing measurement and tools and parameters and assays to go after that value, because what we're finding is if we can truly get that value, we can do comparisons between the various products. Here's an example, if this catches up with me, there it is. We're going to do ultrafiltration, and more specifically, we've tested various methods to try and get at that free drug component. One of the best ones that we've had is this ultrafiltration. It's not a panacea, though. It has great advantages, such as being able to monitor that free drug in equilibrium with the various components. But there's also tricks to getting the solid support not to absorb the drug as you're monitoring it. So this is what we've been pursuing, and we also are now doing a tangent of this particular method in that we can use a non-radioactive isotope of the drug or the prodrug, and it will get into equilibrium with the nanomedicine carrier as well as the various components in the blood and in the blood compartment. And we're finding that when in doing so, we're able to make pretty good conclusions and model how that free drug is going to behave and be able, as I said, be able to compare across various nanoformulations. So here's a very good example of, and it almost comes down to philosophy of how equivalent or how similar do nanomaterials and MBCDs have to be. So on the left, you're seeing Abraxane, which is paclitaxel, but recently Sorrento completed a bridging study, an in vivo study, to reference and compete with Abraxane. So the way this was pursued is under the 505(b)(2). But as you can tell, one is a protein-bound form of the drug, the other is a polymeric form of the drug. So how do you compare those types of apples and oranges? Well, to do so, they went in vivo. The FDA required them to do a comparative study, a bridging study in vivo. And as you can see there, the PK turned out really nice as far as the total drug. It remains to be seen, though, whether that's going to be observed in vivo for efficacy, because as you can see there, they're monitoring the total drug, not the free drug. Those two formulations have different release profiles. So what happens in vivo at the tumor site remains to be seen. Hopefully it will be fine. What's interesting, though, is that the originator of Abraxane now purchased this particular formulation. So it will be a great case in the clinics as both of them are compared. Okay, if the one thing I want to make sure that you understand under NBCDs are the C in NBCD stands for complex, and nanomedicine being a subset of that is certainly as complex. We have terrific ways to monitor and measure the various PCC parameters, size distribution, and so forth. But as you heard several speakers say, the process is the product. As we saw 2 years ago with Doxil, you can't just follow a recipe and go forward. I've seen and heard several comments about making standards. Well, from our perspective, those standards help for calibration of the instrumentation set and or the precursors as opposed to the actual formulation, because it's the lot release criteria for GMP is now defined by boundaries. How much pegylation and stoichiometry of the various excipients do I need to be able to compare or to have that same product? So I think it's just my opinion, and my opinion alone. It's unlikely that we'll have reference material standards for a specific formulation, but certainly for the components as well as the methods to get there. I just can't see where something like a monograph will play into these very NBCDs because they're just exceptionally complex. They will need new methods. We're, as I said, aggressively pursuing methods to monitor the analyte, the free drug, and we believe if that comes to fruition, and we're being funded by FDA to pursue this, that we'll at least have a method a common denominator to be able to make comparisons between the legacy and the follow-on. At least that's what we're hoping, to make everyone's job a little bit easier so we can, in fact, compare various formulations. So with that, I've got just enough time for questions, and I don't need that. Okay, thank you.
Okay. Thank you very much, Scott, for this presentation and view from kind of different institution, but I think a very good one, which is assisting in a lot of aspects. Again, burning questions, please, and give your name so that we know who and where you are.
Avi Schroeder from Israel. I want to thank you also for a great talk. And Scott, I did want to ask you, so we've seen the escalation in publications in the field of nano and nanomedicine, which creates many times one paper that says A and the other one that says the anti-A. And do you see your body, which is unbiased, as maybe a new type of judge in the good manner of actually helping the FDA see what's real and not real? Because when the FDA, at least in my eyes, gets a lot of information, sometimes it's hard for them to make a decision that's unbiased because of all the information out there?
Yeah, so great question, Avi. So we are a formal collaboration with the FDA. They have unfettered access to generating, which includes confidential information. We just call it like we see it because we are a third party. We represent both the sponsor and we represent, or we have that formal collaboration with FDA. So you're exactly right that sometimes a specific method will be pursued and touted as the best one, and then other times it's no, you can't use it. I think that falls back to how the dogma is that each nanoparticle is unique, and a specific portfolio of instrumentation that adequately addresses the boundaries, and the PCC for one product won't work on another one. One may be hard, one may be soft. The kinetics of drug release may be different between the two. So I don't know that I would just say and mandate that you have to use this specific instrumentation or this specific assay. Yeah.
Okay. I think we have another chance by the end in the
Thanks.
debate. Thank you, Scott, for your presentation. And